All right, good afternoon, and thank you for joining us here today. I want to take a moment, though, to thank CanMed and their entire team for putting this conference on, and also the time for our panelists that they're spending with us today. I'm the moderator today. My name is Michael Straumietis. My friends call me Big Mike, and I'm the CEO and founder of Advanced Nutrients. We manufacture a complete cannabis growing system consisting of fertilizers and additives that optimize every stage of the plant's growth from seed to senescence. All right, so today's panel is going to be about the role that nutrients play in yield, cannabinoid output, and plant vigor. Now, before we get started, I'd like the panelists to introduce themselves.
So I'm Karl Ravet. I'm a PhD in plant molecular biology, and I worked on plant nutrition for more than 15 years, specifically focusing on iron, metal nutrition, iron and copper specifically.
My name is Afia. I am the Chief Science Officer at Cannasave. Been in the cannabis industry since about 2013 or so. Been a while. I've been instrumental in setting up a couple labs across the state. Currently at Cannasave, pretty much was a growth leader in the space in California. So we've been able to do that successfully. Also co-owner of a consulting company, but I'll stop there.
Hello, I'm Allison Justice, and I am a plant scientist. You'll probably hear it in my accent, but I'm from South Carolina. After graduation at Clemson University, I went out to San Diego. First time I've been back in quite a while, in SoCal that is. But I worked for a vertically integrated company in San Diego where we grew indoors, outdoors, greenhouse. But currently, and for the last 4 years, I am back in South Carolina where I have a hemp company that's vertically integrated and also a research company that we can get into more later, Hope mentioned earlier.
I really want to talk about the research company. So in the meantime, Hope Jones, I got my PhD at the University of Arizona. Plant Sciences was my minor for my PhD. It was my major as an undergraduate. My PhD work was actually in molecular cellular biology, and I was doing cancer research. And I also worked at NASA researching how to grow plants in space. And then I took a period of time off from the hard sciences for a while until the cannabis industry came calling. And then now I'm back full circle, actually, which is very, very cool because this is where I'm the happiest, is where I actually get to work with people and work with plants. So I get both of my my two favorites, but it's really about, to me, I see it as a playground for us. And I cannot do it without the knowledge of the people like you, Big Mike, and other, like my partners I work with, because I did not come from the industry, from the black market days. And there's so much to understand about this plant that it's one of the reasons actually why I come to conferences all the time is to help me be better at what I do by learning about these strains and these genetics. I mean, it's just, it's been a long road for you guys.
Been a long road for a lot of us. The first question I got for the panelists are, what are some of the commonly held misconceptions that you found out about cannabis in your research? This is a great topic. So what misconceptions about cannabis in your research that you found?
You can start,
sure,
yeah.
Karl.
So, I mean, I don't know if it's a misconception, but it's at least a mislabeling. When we talk about micronutrients, usually people think they're not essential, or, you know, not as essential as nitrate, phosphate, and in fact, they are as essential as any other nutrients. They are characterized as micronutrients because they accumulate only at a certain level, a few ppm in the plant tissue, but they are more than essential. And if I can take iron as an example, because I worked a lot on this nutrient, iron is utilized in directly or indirectly any reaction in the plant. The two main reactions involving iron that are critical are respiration and photosynthesis. These two processes are just the energy plant in a cell. And without this, basically, there is no plant development. And just to capture one molecule of light, one quantum of light, and to transform it into a molecule that is useful for the plant, the photosystem, which is a complex that captures this light and transforms it in chemical energy, requires more than 20 atoms of iron. And any missing iron atom in this complex will completely disrupt the light energy capture. For the respiration, the same. If the plant doesn't have enough iron, the respiration will be compromised and the plant development will be dramatically affected. So I think one of the misconceptions is that micronutrients are not as important as macronutrients, but micronutrients have a macro function. And actually, iron is a very example for that because life could not have evolved without iron. Iron is utilized in all the organisms. Nature hasn't found any ways to supplement iron. There are a few tiny examples where copper or other metals could replace iron in one protein, but life is sustained based on iron biochemistry. So it's more than a critical nutrient.
That's great. I'm really here on the analytical side. You have this great wealth of knowledge, experts that are sitting here more in growth. But one of the ones that I've heard is the correlation between your cultivar or your strain color with the potency. And I don't think this really comes from the seasoned growers. I think this is more of a layman's general public thinking perhaps, when you get those vibrant purples, hues, all those colors, that generally it's higher potency. And of course, being in the analytical lab, I get to, usually prove them wrong all the time. As a matter of fact, I've been seeing the reverse. I have a couple of hypotheses about that, but I've sort of been talking to some folks, and really, what's the case around, certain colors when you have your harvested plant having certain colors and having lower potency? Initially, my hypothesis was that it has to do with sort of the ability you're driving your potency. Well, you're driving the plant to produce more anthocyanins, usually responsible for the colors. There's some other things that could be responsible for that, flavonoids and whatnot. But chances are you're pushing the plant to focus on another part of its energy and something else, rather than making high potency THC or increase in trichome size or whatnot. So I get to hear this a lot, and generally when we get that in the lab, you see that the lower potency ones are also colorful. And I think Allison here will be able to tell you a little bit more later about, why that may be the case. But it's just an interesting misconception that I hear out there.
Okay, so I would say my biggest surprise being in the cannabis industry is the application of phosphorus. And so, going back to grad school, reading by the book, yes, phosphorus is needed for flower production. But if you're thinking of other crops that, other plants, of course they need phosphorus. It's one of the essential nutrients. But, thinking back in grad school, we applied 10% with, parts per million, with 20 parts per million being kind of that top leveled out, that's the extreme. Well, when I got to San Diego, 2016, the company I was working with, they already had a cannabis nutrient company that they were using, not yours.
We'll change that.
And so, the label seemed vague, and me being used to actually taking individual salts and making my own fertilizers, I said, okay, well, I have to figure out what's in here, see what's very different. And they were applying 100, up to 200 parts per million phosphorus in flour, which is just mind-boggling. Because, if you think of field crops, what's our biggest pollutant? Well, it's phosphorus. And so even though, the mindset in cannabis is be efficient, be sustainable, there's actually a lot of pollution happening. That extra phosphorus that you're applying is going right down the drain. Brian Whipker at NC State has done some great work in nutrition. It's for hemp, but I've got to play on both sides, and it's all the same. But he's got some excellent papers out there showing, well, if you get over 15 parts per million phosphorus, you're not doing any good. You're not getting more yield, you're not getting more potency. You're just wasting money and polluting the planet. So I guess my takeaway is, a lot of the misconceptions of, well, yes, phosphorus is needed for flowering, let's pump the phosphorus in there. It's kind of backwards, because as you know, there's lockout, there's all sorts of other problems. You start applying more phosphorus, well, you're going to block the uptake of certain things, specifically zinc. And what is that causing as a problem down the road? So know your nutrients.
Yeah. Yeah, for me, as far as misconception, I was shocked. One of the first presentations I gave about 6 years ago, I was talking about virus, and I showed a picture, because I walked into it at the grow that I was running, and I was walking down, and so when I'm giving this talk, the thing that shocked me was nobody believed that cannabis could get virus. And so, and still to this day, I might hear that occasionally, but it shocked me. And what was actually even more concerning was with our own staff. When I found it, walking down the row of, in this particular facility, the moms were in the middle. It was one large bay in a greenhouse, and so the moms were in the middle, the clones were on each side of the mom, and then the veg plants and the teens and the early veg were over off to the side. And I'm walking down the rows, and I just was stopped, and I said to the guys, I said, well, what do you think's happening here? And they said, CalMag. And I get that answer a lot. Oh, it's a CalMag problem. And you know what? It could be. It could be. But then that's when we engage some of our troubleshooting mental checklist. Okay, so if it's CalMag, the other answer I got from the other kid that was there, and he was in charge of watering, he's like, oh, it's a pH issue. And I said, it could be, but do you measure your inputs and outputs? Mental troubleshooting checklist question number 1. If it is a certain nutrient deficiency or toxicity or cation competition, and all the plants in this row or in this room are being watered and fed the same, would you expect only to see it randomly in these 3 plants in different rows, but they're close together? Those kinds of misconceptions, and everybody goes right to the nutrients in their responses when it came to disease. That I think that's one of the reasons, again, this is my 2 cents in theory, that the disease status is so bad, because we're so good as an industry at sharing everything or wanting to have everything that is just killer and killing it at the cups, but we don't quite understand our nutrients and then the troubleshooting behind that things get away from us, and then now it's a problem that we have to deal with at a national or international level from a disease status.
Go ahead.
Oh, I was just going to add to that. That's a great point. Real life experience is we had a client bring his plant to the lab. It was sick, and they thought it was nutrients. So obviously we run HLVD and some other beet curly top viroid on it, sort of having a little experience around that. And sure enough, it was, HLVD that was infecting his plants. I mean, it was pretty bad, even on the dried cured flower generally, which you don't test for. We did, and it was evident that it was there, and it really wasn't a nutrient situation. So
go right to the newts.
and also, Hope did a really great paper for us on HLV, and you can find it at advancednutrients.com/HLV. Karl, I have a follow-up question for you about iron. What forms of iron can translocate, and which forms of iron cannot in the plant?
Okay, yes, iron exists in two forms. The ferric iron, which is oxidized iron, Fa3+, and in a reduced form, which is called ferrous iron 2+. And I think this is basically the source of its use in biology, because once you have a protein that binds iron, this protein now has the capacity to take an electron from another molecule and to transfer it to another molecule by changing its valence between iron 2 and iron 3. So iron loses an electron to give it to a molecule. And this is how all the oxidation reaction works in living organisms. That's why it's so important, in fact. And so it also exists in the soil in these two forms, iron 2 and iron 3, much more as iron 3 because iron is easily oxidized by oxygen. So there is plenty of iron on the Earth crust. It's very, very abundant, but unfortunately it's all oxidized because of oxygen from air. So it's like basically rust. And this is very unavailable to plants. The plants like cannabis, because there are different types of strategies depending on the plant, but plants like cannabis used to take it up as iron 2. So it has to be in its reduced form, which is basically very poorly available in soil. So there is a very low amount of ferrous iron. So the plant have developed some system in order to acidify the root area in order to basically transform this iron 3 into iron 2, making it soluble, and then a very specific transporter, a kind of channel on the root cells can take up specifically this iron 2. Now, cannabis has also a secondary system that can take up iron 3, but iron 3 needs to be chelated to different molecules, organic molecules. And as a complex iron 3 plus organic molecules, these can be taken up by the roots by a different transporter. And then once it gets inside the plant, iron is so reactive by changing taking electron from other molecules, it's actually a very dangerous element to deal with. So it has always to be chelated into the cells in order to be less reactive. So it is basically transforming across the plant and translocated to the shoot in the form of iron-free citrate, iron-free nicosaganamine, which are all organic compounds that allow to transport the iron-free into a soluble form to the different part of the plant and to prevent its reactivity, its uncontrolled reactivity with other molecules. And when it gets to the shoot, if this iron needs to get to the shoot to work in photosynthesis, once it's in place in the protein, then it's safer and it will work only in that context and it's not as reactive. But transporting iron from the roots to the sink part is a very, very challenging trip for iron and for the cells.
Great. Also, I'd like to add about phosphorus. Phosphorus carries a lot of heavy metals. So not only are you polluting the planet and throwing your money away, you're also not thinking about the best interest of your patients. What are some of the differences that you've found in the different cultivars that you've worked with?
I can go as far as analytically. Really, the differences have been their affinity, speaking of metals, their affinity for metals. We found that depending on your cultivar, you can feed a whole, grow with different cultivars, the same nutrients over time, same environment, but at the end up with different residues of those levels, residues of those compounds in the finished product. Took a while to sort of figure out why this is, but really it's simple. It is cultivar dependent. As we know, this plant is everybody knows the bioaccumulator, right? So it generally holds onto these things. However, I guess the rate at which they also release is different. And due to the just general vascular nature of the plant and whatnot, I'm sure you guys are aware, we found that even through flushing, you cannot get rid of them at the same rate through your different cultivars. We've seen in situations, something like cadmium, for instance, certain grower feeds the same nutrients, everything's equal generally, and then somehow one particular cultivar ends up failing all the time for cadmium, and scratching our head, why is that? There's really no clear reason apart from the fact that it just holds on to that a little bit differently. I'm sure there's much more that can be done to find that out, but that's what I've really noticed. That
Has anyone tissue cultured it to see?
I think they're getting that done now after we've gone through exactly why that really is. At the lab, we can only offer so much analytical help, and we'll help you figure that out. But you're going to have to go through tissue culture to get more information.
I look forward to that data.
Actually, it's very interesting because cadmium usually accumulates when iron is deprived in the media.
Right.
Because when the plant feels a lack of iron, the genetic programming of a plant is changing in order to synthesize much more of a transporter to get this iron. So the roots are expanding, and then there are a lot of transporters to be able to capture any atom of iron available. But if there is no iron, the roots are still seeking for it, making a lot of transporters, and unfortunately, the transporter is not specific enough. So when you have a lot of iron transporter in the root system, but no iron to be captured, it will take up cadmium. So one of the main effects of iron deficiency is often a cadmium toxicity, because the system is sucking cadmium. So if you have cadmium contaminant in your soil, in your substrate, then obviously if you have imbalance of iron, you will have cadmium loading into the plant. So perhaps this is something to investigate, because perhaps it's related to that. Yeah.
Happy to get that. So even we're learning up here.
I know, me too.
Absolutely.
Taking notes.
I guess for me, the most evident nutrient deficiencies I've been able to see between cultivars, very evident, very quantifiable, is nitrogen and magnesium. We have hundreds of different phenotypes in our greenhouses, and, we feed them one recipe. It works for the majority of them, but there's a couple varieties that, it will very evidently show it's still hungry. And it's got the classic signs, of course, of nitrogen, and others with magnesium. And so, for us, to make life easy, because we want life easy as growers, we'll take those specific varieties and just do a top dress. So it gives it that little bit extra boost, but as far as the whole greenhouse, we're able to just have, our certain stock tanks of just a very easy one feed.
You have with your work on calcium, could you talk about that, foliar feeding, the different types of calcium, and when to apply it?
Absolutely. So calcium is one that we feed every single week. The amounts that we feed it seems crazy. It seems like it could be toxic. But a lot of that work comes back from research that my old major professor has done on certain things like petunias and geraniums. And what he's been able to show is, with a certain foliar application of calcium, what we're doing is building cells, building a plant that's tough. And so whether that's the infection of fungi, whether that's insects, what we're doing is building those cell walls. And so, calcium in many forms, if applied foliarly, thank goodness, it's not toxic. And so what we've shown in petunias and what we're seeing now in cannabis is that you can apply rates every single week of 500 parts per million, which is a lot. If you think of what you might apply through the roots, it's 50, so significantly less. You're not going to get toxicity, you're just going to get uptake and really build that plant's natural defenses. But the one thing that is extremely important to understand is that calcium is not just calcium, is attached to something else. And so the common forms are calcium nitrate, and so nitrate, nitrogen, and calcium chloride. Basically what you put down when it's snowing to make you not slip. But it's getting more and more popular for people to talk about crop steering. It's a huge thing that people have known for forever in tomatoes. Certain nutrients, certain things will make a plant steer more vegetative, and certain ones will make them steer reproductive. And so I guess the takeaway here is if you're growing a vegetative plant, and whether this is in propagation or as mother stock, you want to apply calcium nitrate. This encourages that vegetative growth. This applies nitrogen that maybe you're lacking from bottom feeding, but that is the form you want to apply. If we think about when switching to flower, if you continue with that calcium nitrate foliar application, your buds will be funky. They'll stretch out. The plant's thinking, I'm trying to flower, but this nutrition regime is making me think that I'm wanting to stay in the vegetative state. So the takeaway here is, go from calcium nitrate into flowering where you're at calcium chloride. Same thing, up to 500 parts per million calcium, and so far it works very well.
I have a question about that. So when it's going into flower and you see those colas starting to stretch, is that a similar to the monkey tailing, which we know is more of a light stress signal? So how different is this from that? That's pretty cool.
So I've always called it foxtailing.
Foxtailing, that's...
I like monkey though. In my experience, what I've seen with that is it's kind of just the tip of the flower that's making this weird curl and elongation. Whereas when we apply calcium nitrate, the whole flower is just kind of expanding.
I got you. Yeah.
And even though in our research we see a higher yield, that's great if you're just going for extraction. But if you're going for aesthetic, you're not going to sell that. It's very ugly. So I think it's a little bit different, but same concept, which is a whole nother rabbit hole we could jump down. Specifically, the CRC is Cannabis Research Coalition is going through lighting work, and what we're seeing. Everybody knows the 2-week stretch. You flip to short days, and there's a stretch. What we're seeing is that stretch is based off of cultivar dependent, but also based off of how many hours is your short day. If you switched directly from 18 or 24 into 8 hours, it doesn't expand as much. It doesn't stretch as much. If you go straight to 12 hours, you're going an extra third of the plant. So, still tons to figure out, but that's kind of what we're seeing on that side.
That's dramatic, like from a yield perspective too, wouldn't it?
Absolutely.
All right. One of the things, a common theme I've heard growers say fertilizer is fertilizer. And actually it's not. The inputs and the forms of those inputs, EDTA, DBTA, or EDDHA, let's say for iron, matters how the plant is going to uptake it and use it, calcium nitrate versus calcium chloride in flowering. So would you agree with that?
Yeah. Something interesting is now with the knowledge we have regarding how genes are used in these different nutritional status. We know, for instance, during iron deficiency, what genes are on in order to synthesize protein to take more iron. So I think with all this transcriptomic analysis, we can have a diagnostic basically on the nutrient status of our plants for calcium, for iron, for nitrate, for any nutrients. So now we have the access not only to look at the plant or look at the final yield. We can basically in real time, pretty much a couple of hours, know exactly what the status is in different developmental stage in order to optimize basically the whole nutrient program during the whole development of a plant. So I think now we have very powerful tools to improve our formulations. And
You're talking about looking at the molecular level?
yeah.
Right. All right, next question. So what in research or anything that's developed in cannabis gets you guys really excited?
I'm excited about the capability that we have today to transfer some known mutations that could potentially make a cultivar much more nutrient efficient just by taking mutations that are known in different plants or in a specific cultivar that you don't have access to, or you like these attributes but you don't like all the other attributes. So to be able to take that specific gene variant and to copy it in your own cultivar of interest that you want to improve. And using genome editing, which is a brand new technique to make some genetic modification without being called a GMO, is a very powerful technique that is opening a lot of avenues in order to develop elite varieties that will use nutrient in a much more efficient way.
That's great. Analytically, I'm really excited to move away from THC. And then, thank you. Yeah, don't take that the wrong way, but the focus on THC is a huge issue, right? The plant provides so much. I mean, if you were in the medical practicum or any of these talks, you keep hearing about whole plant medicine and how all these other components of the plant help, whether in just passage into the bloodstream and whatnot, but I'm excited to refocus. I'm really actually right now focusing on flavonoids. I mean, there's much more to be learned about flavonoids, but in the nutraceutical industry, flavonoids have been known. I mean, you can buy vitamins that are strictly flavonoids. They're anti-inflammatory response or activity and whatnot. There's so much more, and there's plenty of them in the cannabis plants, including, cannabis-specific ones like canflavin. So that's something I'm really doing a lot of work on currently to learn more. And we know that nutrients and the environment have an effect on those compounds as well. So as much as, nutrients have a huge impact on your THC output and the rest of the environment, so along with the other components that are medically useful as well. And I think we really overlook that. What I've found is that those flavonoids actually differ quite a bit from cultivar to cultivar. So from seeing canflavin at high residues in some high THC components, separation between the A and B form, and 17 other flavonoids that I've seen so far is really interesting, and I'm excited for the industry to, at some point, hopefully in the near future, start to refocus on the whole plant medicine rather than just, high THC.
So for me, it's photoperiodic response. And so, now being in South Carolina, I'm outdoors, and it's not as easy as 18 hours, 12 hours. I have to understand at my latitude, when will these when will these plants initiate flower? When will they finish? When do I have to get my regulatory test where I don't get charged for a federal crime for 1% THC? I have to understand this backwards and forwards. And so, what I'm seeing with the varieties I deal with is that, maybe not all autoflowers are actually truly not photosensitive. What I'm seeing is, well, if you take that autoflower and you put it at 18, 19, 20 hours, it's gonna stay vegetative. And so that means it's not truly an autoflower. Well, with a plant like that, it's special. And if we understand the photoperiod and we understand how to work that plant, it's special. So just for example's sake, you have a plant, you take clones from it, you keep it vegetative, you plant it in your greenhouse. You can't afford light depth. You don't want to pull black cloth because it's terribly annoying, and you raise the humidity and all sorts of other problems. Well, instead you grow I'm coining this term, I don't know, semi-auto, where it is
Trademark it.
it is photoperiodic, but very different than what we're used to. So instead, you're able to take clones from this plant because you kept the mom at 18 hours. You plant a greenhouse full of it. All you have to do is put up, very inexpensive LED string lights all year round, because nowhere in the U.S. gets over 18 hours of light. You keep those plants vegetative. You get them as big as you want. You turn those plants off and they flower year round, back to back to back. And so it's just, understanding how light affects those different varieties. I think we can really take advantage of that, really save, for the grower, save time, save effort, and do what we need to do, but easier.
It's hard to follow. So for me, I think one of the pleasures has been seeing more and more scientists enter this industry and working with the people who provided this industry to us, right? So we're all here because of everybody before us, and there's nothing more amusing to me to see somebody who's been in the industry growing for 30 years start talking like, or already like talking like a scientist, but not getting the credit, starting to get more credit and still talking the talk, and then more scientists talking like bros. It's been wild because it's this merging of these worlds that should have always been together, that we now are able to start doing the research that we should have been doing for decades and decades, and we can start understanding regional differences in our genetics, which is something that has been very hard. I know with doing hemp production and selling clones or seeds nationwide, how is any one company going to have all that data to be able to speak to you in Washington versus somebody, or even really truly understand the genetics and have the confidence behind the genetics to talk about that? So those are the things that actually has been nice to see.
Well, what gets me excited about the future of cannabis is marrying three technologies together, and that is, number one, genetics, then the growing environment, and the fertigation program. There is technologies out there with crop steering that are more and more coming out all the time that will actually give the grower godlike control in an indoor environment over the plant. And that keeps me up at night and gets me excited because once you put those three together, you can truly optimize that plant to its true genetic potential.
Can I add one more?
Yeah, sure.
I want to add one more that I just, because Justin and I were talking about this earlier before our panel, and I think probably what motivates us as a business, and I am kind of speaking for you here, but you said it too, so that's why I'm making sure to dish out the credit, but it's something that has always been a part of my model and do currently with everybody, but we don't talk about it enough, and that's actually working with breeders. So as a tissue culture company, yes, we can get into the idea of doing that biotech kind of breeding and the hybridization and stuff. Okay, we'll do that, but why would I waste all my time only doing that and not actually working with these breeders that have these amazing strains? And why would you want to work with us? And so, my argument for that is because when you put your brand out, it needs to represent your brand and that plant or that cultivar and its best possibilities, its full genetic potential. And right now, when people are buying clones from nurseries and they're diseased, is that representing your brand well? I mean, why not work with groups like us where we can actually, it's your brand, it's just we're producing those clean stock plants for your brand, through your brand, and we do everything through licensing, royalties, MTAs. Everything should be on the table and be an option. And that, to me, is going to be the most exciting next 2 years for me personally, going into a new lab in Arizona, being able to do that, because no one group is going to have all the flavors of the rainbow. There's so many good strains out there and breeders who are not getting recognized because they may not be the big, 10 seed packet going for $1,000, but it damn well should be. And so we can get more of that vegetative material out with working with our groups.
I agree. There's kind of a conflict between, let's call them the legacy growers, and what's happening now in the marketplace. And a lot of them aren't coming out of the woodwork, and they absolutely should. They have absolutely just killer strains. Some of these things are giving 42% THC. Who knows what minor cannabinoids they could have in there? And you're right, there should be licensing deals between companies and growers, and they shouldn't be weary and afraid of, as they call them, the suits.
Yeah, I do think that there's not been any favors done by previous, mistakes out in the industry, and so the best that we can do is just say, look, this is something that we can do it right, we can be transparent, we can do things, but damn, get out there, get your brand in the market, and let's represent it in the best possible way, and it's a win-win for everybody.
It is.
Yeah, and it's doing you justice to your work. It's not trying to be stolen from you, so thank you for pointing that out.
Absolutely. So, What grower-specific advice do you have for the audience? Grower-specific advice for the audience.
I mean, if we talk about nutrition on the nutrient formulation you are using, either you are using a commercial formulation and you just have to follow the label. If you start playing with nutrient, it's very difficult. I would warn you that as soon as you change one nutrient quantity, perhaps in order to find the optimal dosage for iron, let's say with one basic formulation, you identify 100 micromolar to be the optimal concentration of iron. If you start then to change nitrate and phosphate in the original formulation, now the optimal amount of iron will be completely different because all the nutrients, or most of the nutrients, are interacting together, and we don't actually today understand exactly how they interact together. So trying to trick formulation on your own is a very careful approach, and I would definitely advise you to not do that, or just to do it scientifically with the proper approach, but not just gamble on your crop by just changing one nutrient, because just 1.5 more iron as you should, you kill your plant. So trying to optimize nutrient formulation is a professional job, I would say, and you have to have the capability and many experiments. If you do it in your basement, then obviously you might have bad surprises. So this would be my advice. Don't play with nutrients as you wish.
Yeah, going off of that, it's really great. In the laboratory, we generally have growers who want analysis of their nutrient, either what they've premixed or what the soil around the plant or the runoff. And a lot of times we end up missing sort of the whole picture. So you want to do sort of a mass balance, right? You want to, okay, what was your input? What's left around the plant? What's in the vascular part of the plant? Maybe you take a root and we analyze that. And then what's being flushed out? Take a sample of that and analyze that and sort of see that mass balance. I mean, also making sure that your lab is even testing for the right analytes, like you say, the iron, the various iron oxides and whatnot, making sure you're testing for the right analyte. You can't just test iron. It's like, which iron? And then you need all that information in order to make an informed decision, as he was saying, to change a certain nutrient, right? If you don't have the full picture, you go ahead and change something that perhaps could be very detrimental to your plant. It helps you, especially when flushing as well, right? Whether you're using a premix or you're doing your own ratios, you need to know and get that whole picture. So make sure that when you do ask your lab for nutrient analysis, that you're taking it from several different areas to get a whole picture and do like a mass balance. That way you can make a better informed decision.
I'd say I appreciate these guys. So 2016, which is and is not a long time ago, I'm growing plants legally, and I want to take tissue analysis. And, I spend days and hours on the phone trying to find someone to take it and give me data. And no, cannabis is illegal. No, cannabis is illegal. And so what do? Well, I dry it and I grind it up and I send it off and say that it's tomato.
Good idea.
That's all I could do. And so they send me results back, and I'm like, all right, this is great. Well, usually when you get a report back, they compare it to something. And so they compared it to tomato. Well, sure, it's a great starting point, but there's some differences. I mean, they're going to need something a little different. And so, for years, all I had to go off of, is this plant looks good. Well, what does that mean? I want my nutrients to match this. Well, then you get into, there's a million different varieties, and so it's tough. But I guess my point is, I'm very grateful for hemp being involved has done a lot. There's universities getting involved that are testing thousands of plants, and they're able to give you that bell curve of, well, this is deficient, this is toxic. This is the range you want to be in. It might not be perfect for your variety, but it's at least a range we can work with. And so not to brag on him again, but Brian Whipker at NC State, I think just a month or two ago, has put out new standards of what we should, at least a rough range where we should attempt to be in. And so, having analytical testing, all over the place now is amazing. And so I guess my advice would be use it.
Use it. Hey, so I'm in the same boat. In having previously been CSO for Analytical Lab, first of all, and I'm going to explain why, but dump your IPA, stop using IPA within your cultivation facility, and I'll explain why in a second. And then second of all, get to know your lab. Get to know your lab. I mean, I had this guy Francis call every morning, and he's a chef over in Arizona, and he literally has turned himself into an analytical chemist. And because at 10 a.m., Aaron, who's running the lab, he'd be like, "That's Francis." And he would stop and he would take the phone because Francis would ask questions. This stuff isn't intuitive for anybody, and especially when you're trying to dial in, you want to be able to make a phone call and talk with your lab. And so now that we can be out in the open about what we are testing, I think it's important to know your lab. And one of the things that I learned by knowing my lab is the substrates. I've been having trouble with substrates, soilless, peat core, and it's a variation in if it's going to be what the starter charge is going to be, which, I mean, the whole thing is really important, especially for a tissue culture company, because I use a diluted nutrient solution on my hardening plants. And so if I am, or transplanting my cultures into a substrate that has terrible quality control or has Aspergillus is the biggest problem that I have seen. Aspergillus is that organism that produces the aflatoxins that we have to test for from a compliant perspective. So my advice is to test your substrates, know your labs. I would have never known that the companies were having problems until I spent like 20 minutes in the lab when I was dropping samples off. And I happened to mention in passing, and they said, we've been noticing the same thing. Our farmers and the growers over in Yuma are saying the same thing. So they actually called up the vendor, or some of their vendors, and was asking, and it was the cannabis industry that's putting, at the time, this was probably about 4 years ago, was putting a stress on the supply chain, and they were having to source from different areas, and they didn't have time to keep up with everything. I would have never known some of that behind the curtain aspect if I wouldn't have known my lab and talked with my lab. So then, does will IPA kill Aspergillus? Well, will IPA kill virus when you're in your grow? No, it does not. It does not. So by putting your tools into IPA, you're doing nothing to mitigate the spread of your viral loads, much less you're doing very little to kill really what's plaguing. And Aspergillus is a problem because I would pasteurize my substrates. Then I went straight on to sterilization of my substrates. Well, the reason why, I totally understand now, in a different way than I did before. The reason why we test for aflatoxins most of the states is because Aspergillus can survive high heat. It can survive a lot of chemicals. If you're not actually breaking that cellular wall, you're not killing that microorganism. And it will survive sterilization. So when it survives sterilization and then you moisten it, and in my lab, I put it in a bin with a lid over it, well, now there's no competition. Whew, Aspergillus is, you could see it through the bends, so test your substrates, know your labs.
Yeah, you're seeing that in cocoa and peat?
Yep.
Both, okay.
Yeah, it's terrible. So if you're failing consistently for aflatoxin, look at your substrates.
So I want to make growers aware that there is a shift between macro, micro, and secondary nutrients between the growth and the flowering phase, and it's quite dramatic, and it will affect your yields if you do not get it correct. Anyone else? All right, let's open it up for questions. We're doing a Q&A. We have a few minutes left. Are there any questions?
Good timing.
Yeah, thank you to the panelists for a really fascinating discussion. So question to the panel, what approaches are you seeing, if any, that use beneficial bacteria or fungi to influence plant nutrient uptake and bioavailability? And that's to the panel and to you as well, Mike.
You understand the question?
Just where I heard you right, what bios are used to help with nutrient uptake?
So anything using bacteria or fungi to affect how the plant takes up nutrients or...
So in my experience, we do a bit of contract research for, large-scale hemp production. And in hemp, the prices are... So we have to be very careful about what our inputs are. And so two specific companies, New Leaf, which is a bacteria, and ProMix, which is a mycorrhizae. What we did with them this past season was, you cannot take these products and apply them to 20 acres at a time. Every week. It's impossible. You would make no money. But what does make sense is taking your clones and doing a significant drench prior to planting. And so it's a one-time application, and, we went through the steps, elemental analysis, yield, potency, and so what we saw with these two specific products, there's many others out there, but with these two, and it was consistent with the two, is that it did not increase potency, it did not increase yield. But being in the Southeast where it's hot and crazy humid, and it doesn't get cold enough to kill most of the pathogens off in the native soil, we have terrible pathogen problem pressure. And so, but what it did do is it significantly reduced disease pressure. And so for us, that was a huge win. It wasn't just, well, you took up nutrients X amount more. It was, well, you basically saved the life of this plant. So that was kind of the takeaway we learned. In actuality, it did increase a few minor nutrients, but that didn't equate to what the farmer wants, being money, yield potency.
Thank you.
If I may add one more thing to that, just as you use that, be certain, depending on your state or your regulations, that it may lead to a microbial failure if they're looking for total aerobic organisms and whatnot. In California, it wouldn't, because they're looking specifically for pathogens such as Aspergillus and E. coli. So just beware that it doesn't get onto your final end product.
Thank you. I want to commend you for acknowledging the role of legacy breeders and the importance of integrating them into the industry. And to that end, since we have a fairly big audience here, I just want to announce that tomorrow, last thing in the small room, right before you go off and party for Cinco de Mayo, the impacts of legalization on the cannabis legacy and the need for solutions to create a healthier industry. So I hope some people will attend that. It's really important. And I just want to also put in a shameless plug for my company, Breeders Best. I work with Ethan Russo and Rob Clark, and we're doing exactly what you talked about, bringing in genetics from legacy breeders that have been doing this for decades and trying to find markets for them. So look us up, give us a call. We want to put some genetics out there.
That's a good plug.
So is there a preferred pH to the foliar application that you were talking about with the CalMag?
I would imagine there are extremes that would be detrimental to that, but in my experience, in the broad range of acceptable, there's been no difference. I'm going to rely on you on that question at the extremes of foliar nutrition and
pH.
And pH.
Yeah, obviously, I will confirm that the two extremes are bad. Then after, obviously, for metals, you prefer rather acidic, quite acidic, which is not good for other macronutrients like nitrate, I guess. So it's a thin balance between, having a good equilibrium for uptake of all the nutrients, rather than optimizing, for one. But basically, you will deprive your plant of nitrate if you optimize only for metals. So I think plants have evolved, to grow in the kind of soil pHs, and obviously there is some genetic diversity depending on the type of soil. Plants have been able to evolve within a narrow window, but I think obviously in the extreme, below 4, higher than 8, perhaps it's more difficult for cannabis to grow.
And with the observation of the semi-autos, would this be a symptom of the ruderalis autoflower blending in with photoperiod plants, reducing the effect of the actual production of the hormone based on overshadowing? Or is this technically an attribute that is coming out as its individual statement?
It's a hard one. The varieties I've worked with, it wasn't an intentional cross of ruderalis and a photoperiodic variety. That's a genetics question. That's not necessarily my speciality, so I'm not sure. I would imagine so, but I'm unsure.
In our personal garden, we've actually been able to extend based on light intensity with autoflowers. After a conversation realizing that the ruderalis started out as an undercrop. So basically, as its competitors grew taller, the overshadowing would hormonally trigger blooming. And that's just one of the reasons I was asking if it's a cross between the two in attributes, because I know a lot of us in the breeding culture to have taken the original land races that breed in elongated sectional timings, such as the indicas going 10 to 15 weeks and sativas going 15 to 21 weeks or 25 in some regions, and basically shortening those by breeding in ruderalis plants. I'm just interested to basically see if that's a linkage in the instability we're seeing with hermaphroditic tendencies late in bloom, or if that's something that's just promoting based on our current practices and being away from nature.
So I didn't hear every word. It was a little bit hard. It's a little bit hard to hear.
It is hard.
Basically, I'm sorry. Basically what I'm asking is, are we creating instability by breeding in these too far reaches? And are we seeing that as an attribute to the hermaphroditic tendencies in some plants that occur late in bloom?
I have not been able to correlate hermaphrodism with, I guess, photoperiodic response. I think we're hybridizing just about everything, whether it's photoperiodic response, purple color, whatever it may be. And I think it really goes back to, homogenizing your genetic line. Because sure, one cross of very extremes, you're gonna get a lot of different types of children. But picking that one you like and then, breeding to support those genetic traits you're looking for, I think that's what's gonna stabilize it. Until then, it's gonna be up in the air of what you're getting. Does that answer it?
I appreciate it. Thank you.
Hi guys, my name's Ali. I'm with Cannabis Talk 101. This question is primarily for Mike and Ini, but I'd love to hear from all of you. How important is standardizing nutrients to getting standardized medicine? And do you think there'll ever be a day that we will be able to get such consistency that we're almost at par with pharmaceuticals?
Yes. We've tested practically every competitor out there. We've looked at the cannabinoid profiles, and you can manipulate cannabinoids by macros, secondaries, and micronutrients. And I talked earlier about marrying genetics and crop steering and fertigation. that will give you what you're looking for, as well as lighting and environmental controls, and all the sensors will be involved. You'll be able to steer that plant in many different directions. And there's quite a bit of research that has to be done. We're just cracking the tip of the iceberg when we think about cannabis and all of the different effects that would be the possibilities when you look at the cannabinoid profiles.
Yeah, it's really important. I was once in a production environment. Somebody was pressing rosin, and there were a bunch of new techs there that he had to teach how to, press the rosin out and do that whole process. And he was really good at what he did, but he said that he couldn't really teach that because, the temperature, you have to feel it, you have to feel the pressure that you press at, it's like, okay, I understand that, but that's not how it should be. It has to be standardized. You have to be able to set a certain temperature. You have to be able to set a certain PSI of pressure. These are what SOPs are necessary for. I think as we get legalization or federally regulated, things have to be a lot more standardized. You couldn't just, I get it with the master growers, they've been doing it for years and they know what they're doing, but now the great challenge is to get that written down in a strict, direct manner. I mean, that's essentially what I do. So when I'm, creating a lab or setting up a lab, I couldn't just put everything there and expect to do it all myself. Otherwise, why hire people? I'd have to run all the assays. I couldn't even sit here, right? I have to create SOPs for people to follow, and it has to be standardized. So that has to be the same thing that we have to head towards in this industry. If you're willing to, or if you're going to be able to produce consistent medicine with consistent THC levels, other constituents, flavonoids, whatever, that growing process has to be standardized. Now I get it, there's always situations where there's disease and whatnot that could throw a wrench into things, but think about, the corn we eat, right? It's a very standardized process of growing that. They're not just shooting at the hip about it. So it's going to be very important.
I agree. It's going to be really important. One of the things that I think saves our ass when it comes to competing with the pharmaceutical industry is the entourage effect. And so if we're going to do something that is plant-based and we want to mimic anything like what we have within those trichomes, you can't do it by only synthetics, right? And so there's so much more research to do. So we do stand a chance in the industry, in my mind, to be able to compete with anybody if we're willing to put the investment and training behind proper facilities, and HVAC is one of them. It's the biggest mistake that I've seen in almost every facility that I go in is just a crappy job of HVAC or dealing with what they have. Then it comes down to tracking the data, and we need a baseline of genetics. So one of the problems that I see with so much of even the current literature that we're dealing with is until we have markers and we know a strain is what it says it is, we have no baseline as an industry. Now, we have years of anecdotal evidence. When you work with your plants, regardless of what its name is, you know its growing habit. How do you replicate that on a commercial scale? And then to be a commercial scale, every commercial scale industry deals with new genetics coming in on a regular basis. We are so tied to our genetics. I have clients that will not get rid of a diseased plant because it's the last one that they have and it's irreplaceable. That is a terrible way to go about business. But we care about our genetics like they're our children and our pets. And that's okay, but then we need to learn to maintain them so we have healthy plants and children, right? So there's nothing wrong with our way. We just need to meld some of additional ways and finances into how to do that on a commercial scale, and we can compete with everybody.
That's a great question.
All right. Well, we've come to the end. I want to thank our panelists today and our audience as well. Thank you very much.